native snails, but no native slugs. Some of the slugs that you find, slugs and snails that you find, can be very damaging to agricultural crops. There's even cases where shipments of agricultural goods were sent to the continent and sent back because they found gastropods that they didn't want there. So that's one way that slugs and snails affect productivity in the islands. Another way is that they also carry diseases or parasites. They carry diseases like rat lungworm, which has become quite a topic lately. You also have items like the garlic snail here. Some of you probably don't know this, but these are actually carnivores. They eat other snails, which is a big threat to the native snail population, and when the native snail population goes down, that's also a food source for the native birds too, yeah. Gotta put the clicker quite closer. So the three questions I came up with for my project was to, number one, figure out who's out there. Number two, figure out do the canopy types within our project area make a difference, and what kind of plants do they prefer to consume. So our plot was in the Mountain View area just off of Stainback Highway. The Hawai'i Timber Management area was relatively completely bulldozed in the 1940s and replanted with plantation-style blocks of hardwoods. So what we did was we found a little intersection that covered 3 different forest types, and that's where we set up our points. We were basically at about 720 meters in elevation. The Hawai'i Timber Management area runs from 125 meters elevation all the way up to 975 meters elevation, with annual rainfall between 500 centimeters per year in the lower sections up to 380 in the higher sections. We were at about 720 meters. So what we did was we focused on 3 of the top 4 tree types: eucalyptus, tropical ash, and Queensland maple. And this is how they break down within the 4,800 acres, 8,500 hectares. That is the Hawai'i Timber Management area. Within our sample areas, we also found lots of native plants, alani, lots of hapu'u. We found a single lu'ulu. There was also massive wahia in there also, along with lots of uluhe and ieye. The table you see is from an inventory from 1997 done by Fall. So the first thing we had to do, cut some trails. And what we did was basically we set up 2 transects within each forest type. Each transect was 200 meters long with 20 individual points on each transect that were 10 meters apart. This is where Naoli was doing her tracking stations. And off of these transects, 10 meters off to the left to the right, were 10 by 10 plots that I set up for my sampling. The points for the plots were randomized, and that's how we got the locations. And there was also a 75 meter from the other forest types, so nothing else was around there to interfere with our project. The first step was to do a above ground time search. We did a 3-man team search for a total of 13 minutes and 20 seconds. That equates to 1 person working for 40 minutes. And we took counts of gastropods from everything from the ground up to 2 meters above ground. The next step was to go through the ground and leaf litter and search for 6 minutes and 40 seconds. Again, it's 3-person team, totaling 1 person searching for 20 minutes. So 40 minutes and 20 minutes in each plot. We searched for 1 hour of max. The next section we did was beer trap sampling. Basically what we did was we found, it's common knowledge that slugs enjoy beer. They like the smell of the yeast and they're attracted to it. So what we did is we created 8-ounce mason jars with a 2-centimeter hole in the lid, and we filled it halfway with beer, If you notice there, we actually buried it up to the lid, so that it makes access easier and prevents tipping. And because I'm working with beer in the forest, some of you underage guys probably think it's cool and stuff, but you know, I just want to let you guys know that alcohol is bad for you. Okay, so we moved into the greenhouse, and what we did in the greenhouse was we started an experiment with 30 different pots that we created by hand to enclose plants and gastropods together. So the first thing we did was to create the pots. The first step was to seal the holes on the bottom with screen and hot melt glue. And we used wire to make a crown-shaped frame that went into the soil to support the screen that closed it off. And we used rubber bands to cinch the bottom so that they couldn't get out. Within each pot, you see momaki, Clydemia, ohia, and strawberry guava by the city of Kapiolani. So what we did once we got that settled, we let them sit for about 3 days so to make sure that all the plants were healthy before introduction of the animals. We measured the heights, we counted the leaves, we measured the animals, and we sealed them by folding the tops and paper clipping them closed. And we took all 30 pots, 10 with slugs, 10 with African snails, and 10 controls with just the plants. We put them all on the same table, set them up under sprinklers, and ran the experiment. So let's get into the results. What did we find? Question number 1. Well, this is what we found. This ugly guy up here, Nemanium, Striatum, Arion, Limax, Duracis, Reticulatum. Tornatillus is a native. Succinea thunumi is also a native. The most common ones that we found were the Arion and the slugs, and snails were the Succinea. This one is actually on a Hawai'i branch right there. Oh, and Limax maximus was the winner of the beer trap competition. There was more Limax in the beer trap than anything else. This is the time search totals. Basically about 117, 111, and about 106 were the total counts of what we found snails and slugs per forest type. But we wanted to get a little bit more details, so we looked at where we found them. And we noticed that above ground was significantly less than on the forest floor. So we broke that down even further into slug and snail abundance by habitat. And this one was really good because it showed a significant difference between forest type and habitat type. So there was a difference as you go along between slugs above ground and slugs on the forest floor. So basically what you're seeing here is the number of individuals per 10 by 10 meter plot is highest on the ground and above the ground for slugs. And in snails, it's the opposite. We find snails above ground more than we do in the forest floor. And there's no real difference. Oh, no, yeah, this one is no significant difference between the forest types, but there is a difference between the habitat. Then we move into our greenhouse results. We'll start with the momaki, because momaki was highly prized by the snails and slugs. This graph here is actually percent of survival. The end value for this graph is 10, so each 10% drop is actually an entire plant getting consumed. So you can see here from the snails, on day 1 they ate 2 plants, slugs ate 1, by day 6, nada. And by day 13 the slugs caught it. And in fact, they liked momaki so much that we even had like one come in and join our experiment. During the control resting period, when I went to go and put the animals in, I opened up one of the control things, and one of the momakis had gotten eaten by a tiny little giant African that came into the greenhouse and climbed up into my pot and ate my momaki. So yeah, they really like it. And secondarily, we started noticing after the Mamaki was gone that there was some damage to the 5V. We didn't really run it long enough to see if it would be significant, but we did have a test pot that I created to make sure that our seals would work and they wouldn't get out. And as an experiment, I just threw in some 5V in there, and by the time day 13 of the trial came, that pot was probably down to about 10%. So, in conclusion, forest snails like to be in the trees, and forest slugs like it in the ground. The most common snail is going to be the native Succinella tanumi, and the most common slug is Aria intermedius. Omaki seedlings attract the most damage. As she's going to present later, she's going to also show you that there was also slug damage to native seeds that we used for her project. So not only are they consuming plant matter, but they're also consuming the fruits and preventing the plants from completely destroying. And so I'm going to hand it over to my partner, Oli.
Hello everybody. So my project for this summer internship is fruits and seed removal by vertebrates, non-native vertebrates. Invasive species is a really big effect, or they cause a really big impact to our native Hawaiian forests. Well, not only Hawaii, but other forests as well. Some of these invasive species are feral pigs, rodents such as rats and mice, and also the mongooses, as you can see that. Anyways, they can destroy native plants, and also they can disperse non-native plants. So my research question is, my first one is, are native and non-native plants, fruits, are native and non-native fruits and seeds removed by vertebrates, non-native vertebrates? And my hypothesis for this is that fruits and seeds will be removed by 4 main vertebrates, such as rats, rodents, mice, feral pigs, mongooses, and caliche pheasants. And for my second hypothesis, it's rats will remove the most amount of fruits from these from my treatments that I talked about. My second question is, how do removal rates differ among forest types? And hypothesis for this is that the highest fruit removal will be in the forest type with the highest rat activities. Because as you can see, my first hypothesis is that rats are going to be the main impact on my treatments. So the three areas that we worked in is like what Martin said earlier, is the tropical ash forest, the eucalypt forest, and also the Queensland maple. My first animal census method that I use is these tracking tunnels. As you can see, these tracking tunnels, they are to compare the rat activities or also small little mammal activities such as cats and so forth. And in this cardboard looking tracking tunnel, it has this black, I don't know if you can see, but this black print ink on the front part of the tunnel and in the back. So what the rat does, or the cat does, or the mongoose, any of those kind of small vertebrates, mammal vertebrates, is that they walk inside this little box. They step on the black ink and then they grab the fruit, or they start chomping on the fruit. So what this helps me do is it leaves their foot marks on top of these boxes. So we can determine what is in this forest, or what's going on, what's here. So you'll see my results later. The next experiment methods that I use are these 3 different types of treatments, which are the total exposure, the exposure control, and the open exposure. The total exposure is only to allow non-vertebrates to come and eat the seeds that I put in these exposures. So no cats, no rats, no mongoose, no, all those kind can't get into this. Only small little creatures such as slugs, snails, and so forth. The exposure control it allows any kind of vertebrates to come in, such as pigs, the big ones, pigs, rats, birds. And the reason why I have an open and exposure control is because the exposure control can have an effect to what the animal thinks about the seed. Like the exposure could scare off the animal because of the caging of it, or it could attract it more. So that's why I have these two different types, the caging and the open. So I've been checking these treatments every 1, 7, and 10 days. The 3 seeds I use are the Kolea, Kopiko, and the Momaki. The next method I use is these motion sensing cameras. And what this allows me to do is to see the different types of animals that's responsible for my seed removals. And the first results that I have is that from the tracking tunnels, I found out that the maple forests tend to have a greater impact of rats tracking than anything else. But the thing is, there's no significant differences among any of the forest types that I got from the tracking tunnels. But a cool thing is that only the eucalypt forest is the area that I found mice trackings on. So it's kind of cool because I was thinking that it's going to be in all of them, but only had it in that one area. And here's our examples. Hold on. So you guys can't see it good, but this is a rat foot. See, it's bigger. This is mice. It's smaller. Smaller foot. And the other one's supposed to show there's no fruit on there, like something ate it. And it has the trackings on it, but you can't see it right now. Hold on. Anyways, my next result is from my open treatment, the one with no caging, just the open. And what I found from the 1st to the 10th day that I checked these seeds, what I found out is that the Mamaki seed was the most fruit that got hit hard, or also known as, it got eaten and taken away with the air than the other two. The Kolea and the Kopiko had the least amount of seeds that was taken. In this graph, for my next results, it shows the Kolea in 7 days. And what I found out is that there's no significant differences among any of the forest types, the 3 forest types that we did. But the thing is, there were significant differences between the total exposure. As you can see, how the graph is, the total exposure in the maple had some fruit removal of Kolea on the maple area. And the reason for that is because in total exposure, only invertebrates can eat the seed. So that's how we know that the invertebrates is what's eating in the maple forest. It's getting darker, huh?
Yeah. Just trying to see if that's better. Is that better, you guys? Yeah. No sleeping.
This is good? Okay. The next seed result that I have is my Kopiko seeds. And what I found out from this is that the Kopiko seed only had a few seeds that the seed was removed from my exposure. So there was no significant differences from each forest type of this one either. For Mamaki, like I said earlier, Mamaki got whacked really hard, which means it got eaten a lot. Oh, come on, Mamaki got eaten really quickly, and there was a significant difference, as you can see, in the ash forest. There's no seeds left in that one. And there's 50% remaining in the treatment, Okay. My next result is in the motion cameras. Those are the 2 pictures that I got from the forest. in these motion cameras, what I found out is that, like I said, rats was the one that got caught on these cameras. And that's so cool because I thought that, you know, my prediction for that was right. But another thing is that I have some pictures of also pigs, mongooses, and colleagues present eating that, and I'll show you guys that later. But here's a rat eating one of my open seeds, which is right there. Here's some cool photos of the colleagues present eating the seed. It's going, gone. So, what I found out that rats' activities from the tracking tunnels was not different among forest types. Rats were the most common vertebrates moving fruits. As you know, I found out from both the seed removal and from the pictures. That's how I found out. And then there was no significant difference among forest types for Kolea and Kopiko, and also the Mamaki. But the Mamaki had no fruit remaining in the tropical ash. That was a really amazing thing for Mamaki. And the thing is, like I said earlier, Mamaki had the highest amount of fruit removal on the TE, which is the total exclusion, which means that the invertebrates consume and remove the most fruit and seed due to the slugs and snails that we found. And talking about that is when I did the day after checking, the first day I checked my seeds, I found out that there were these little slugs eating these mamaki seeds. And then according to Martin, the mamaki seeds was eaten by both the slugs and snails that he had in his greenhouse. I was going to say greenhouse a bit, but his greenhouse experiment. So that's how I knew that these smaller invertebrates are also the cause of these fruits being moved and eaten in the forest. The Kolea had 50% fruit remaining, which also is like that these seeds are also a big risk of having vertebrates, or rat particularly, because I have, like I said earlier, rats, particularly eating these native fruits. And another thing is that the Kopiko had the least amount of fruits that was taken. So they're not as, they're they're minimized on the fruits that's been taken by any kind of vertebrates in the forest. So my overall conclusion is that non-native vertebrates and invertebrates have a great impact in our native forests. Both vertebrates and invertebrates move some of the fruits and seeds of 3 native plants that we put out in our forest, in those 3 different types of forests. And research like this helps us to determine the negative impacts of introduced vertebrates in native forests. So you guys gotta remember that. There is there is non-native vertebrates that is causing these bad damages to our native forest plants. And here too, mahalo.